In this video, we'll talk about cell culture. Cell culture refers to removal of a cell from an animal or plant and subsequently culturing it in a favorable artificial growth situation. For example, you can see here a lung cell has been taken out of the organ situation and kept in a culture. So the lung epithelial cell can be cultured. And in the next example, you can see the meristem or a portion of the leaf is cultured in an artificial media to grow an intact plant. So for both plant and for both animal cells, they can be cultured in vitro or outside of the organ. And that is known as cell culture. So this video is all about cell culture. We would learn about basic terminologies which are super important to understand cell culture. First, we would learn about primary cell culture. So primary cell culture starts with the dissociation of cell from a parental animal tissue. It could be also plant tissue, and enzymatic digestion or mechanical digestion is performed to get rid of the tissue organization and kind of like free the cells from these tissue-like settings. So repeated up and down with trypsin or several enzymes such as acutase, etc. can get rid of these tissue-like organization. Ultimately, it would become cell suspensions. Now, these cell suspensions are plated onto specific media which would support the growth of these cells, and that becomes our primary tissue culture. That means from the organ, you isolate the cell and plate it on a culture setting, and that gives you the primary cell culture. Now, these primary cell culture has different growth kinetics. First of all, there would be a lag phase where there would be no significant growth, followed by a log phase, an exponential growth phase where the cells would grow in an exponential fashion. And lastly, as the cells grow, the media gets limiting, and also there is not enough surface area for growth. So all of these factors leads to a plateau phase. So whenever a cell culture or whenever a cultured cell reach these plateau phase, you would see this is termed as confluent cells. Confluency is commonly used as a measure of cell numbers and cell surface area versus the surface area of the cultured dish, just to get an idea that how much crowded these cells are. If it is too much confluent, then it won't get enough amount of media or necessary nutrients for its growth. So when they're super confluent, then you need to subculture them. And first time you subculture from these cell types, then they would be known as a cell line. So cell line is just a subclone which is derived after the first subculture. So from this culture, you see these cells are now confluent. It's time to separate them into two different halves, and we term it as subculture. So after subculturing, it would be termed as a cell line. Now, cell line, which is derived from a primary culture, have limited lifespan, and as they are passaged, many cells which are having highest capacity of growth, they predominate the culture. So this results in a high degree of genotypic and phenotypic similarity or uniformity in these cell cultured or in this cultured cell population. So let us quickly compare the properties of primary cells versus cell lines. So obviously, I would say the primary cell has a very high biological relevance compared to a cell line. I'll tell you why in a moment. But if you talk about the lifespan, both could have sort of like the primary cell could have like a limited to finite lifespan, where cell lines are generally infinite or unlimited. Now there are many other points where they differ, but these primary cell culture recapitulates the tissue-like organization way more than the cell lines. I'll tell you why. But before that, let's try to understand this process in a lab setting. In order to understand cell culture, we have to virtually explore a cell culture lab. So here is a cell culture lab. You can see the essential components. It should be very clean and it should be tidy. So in this cell culture lab, on the right hand corner, you can see there is a biosafety cabinet. All the actions would take place here. Before looking at the biosafety cabinet, let us look at other components. So you can see just left side of the biosafety cabinet, there are two hoods. So there are two incubators which are important for keeping the cells and they would provide the proper environment for the cell to grow. Other than that, there would be centrifuges, microscopes, cell counter, etc. So there are certain norms that you have to follow when you're working on a cell culture lab, such as wearing a lab coat all the time, a covered lab coat obviously, and if you're culturing something which is infectious or any other serious material, then you should have an additional layer of protection. You should have your proper PPE kit in order to culture the cells. Now guys, what you need to understand is what is inside the hood. These hoods are generally, what is depicted here is a classical BSL-2 hood or biological safety level 2 hood. However, biological safety level 3 and safety level 4 hoods are very different looking, and in other videos we'll talk about that, but not in this video. So you can see there are many things which are associated with cell culture should be placed in the hood, but it should be first sprayed with alcohol and then it should be kept inside the hood such that it is contamination free. Now, while working in the hood, it is always permissible that you wear lab coat and having proper gloves and other protective equipments. This would prevent contamination and also save you from any kind of biohazards.